Welcome to Mythosium, the channel where we explore the ideas and discoveries that transformed the modern scientific world. Today we're looking at one of the most important breakthroughs in the history of biology, one that reshaped medicine, forensics, biotechnology, and genomics. It's a technique so influential that it sits at the heart of everything from ancestry tests to cancer screening to pandemic response. This is the story of how humanity learned to copy DNA at will, and why that achievement fundamentally changed the world. For most of human history, DNA was completely invisible to us, a hidden archive containing the instructions for life, yet unreadable and unreachable. Scientists knew it existed long before they could examine its structure. Even after the discovery of the double helix, the ability to analyze DNA remained extremely limited. Researchers could isolate fragments or identify broad patterns, but detecting specific mutations or amplifying a tiny sample into something measurable was staggeringly difficult. Early techniques required patience bordering on extreme endurance. One of the main approaches used in the 1970s and early 1980s was called the Southern blot. This method allowed scientists to separate DNA fragments, transfer them to a membrane, and detect the presence of specific sequences using radioactive probes. While ingenious, the process demanded days or even weeks of work, involved hazardous materials, and produced results that were sometimes faint or ambiguous. Laboratories were eager for faster, safer, and more sensitive methods. The challenge was clear. DNA is extraordinarily long. Every human cell contains over 6 billion nucleotide bases, tightly coiled into chromosomes. A single genetic variant, a mutation involving one letter among billions, can influence traits, cause disease, or determine hereditary patterns. Detecting such a small change required extraordinary precision. The analogy often used at the time was that identifying a single mutation was like trying to read a license plate from the moon. Scientists could separate DNA into smaller pieces and attempt to isolate relevant sections, but actually analyzing the specific site required an enormous amount of labor. By the late 1970s, biotechnology companies began emerging with the goal of turning molecular biology into practical tools. One of the earliest of these companies was Cedus, founded in 1971. It was an exciting time for science. Researchers had just discovered restriction enzymes, tiny molecular scissors used by bacteria to defend against viruses. These enzymes could cut DNA at specific sequences, enabling scientists to fragment genetic material in predictable ways. Suddenly, the biological world became editable. DNA was no longer a mysterious molecule, but a manipulable code. These discoveries opened the door to genetic diagnostics. For example, sickle cell disease, a condition caused by a single nucleotide difference in the beta-globin gene, became a prime target for genetic detection. If a diagnostic method could reliably amplify and analyze that specific region of DNA, medical testing would leap forward. But amplification remained the bottleneck. DNA samples were often tiny. Each cell provided only 2 copies of the gene under investigation. Genetic detection required millions of copies, but no technique existed that could selectively generate such quantities quickly. Researchers knew that living cells already contained a solution: DNA polymerase, a protein responsible for copying chromosomes during cell division. However, the natural version of this enzyme could not withstand the conditions needed for laboratory amplification. DNA had to be heated to nearly boiling temperatures to separate the strands. Under such conditions, ordinary polymerase would degrade. That meant scientists had to add new polymers manually after every heating step, a tedious process that hindered any attempt at rapid, repeated amplification. The missing element was a heat-resistant enzyme, something robust enough to survive the rising and falling temperatures of repeated DNA denaturation and replication. Fortunately, the natural world had already evolved exactly such an enzyme. In the 1960s, microbiologists studying the boiling waters of Yellowstone National Park discovered bacteria thriving in conditions far beyond what most organisms could tolerate. One species, named Thermus aquaticus, contained enzymes built for extreme heat. Its DNA polymerase, later known simply as top polymerase, retained full activity at temperatures that would destroy conventional enzymes. This discovery would prove pivotal. Once scientists obtained a stable supply of Taq polymerase, the conditions were set for a revolutionary method, a chain reaction capable of making billions of copies of a selected DNA segment in just a matter of hours. The core idea behind the polymerase chain reaction, or PCR, is elegantly simple. DNA is made of 2 complementary strands held together by hydrogen bonds. When heated, these bonds break and the strands separate. If short synthetic DNA sequences, called primers, bind to each strand at positions flanking the target region, and if polymerase is present along with free nucleotides, the enzyme will extend the primers, creating new copies of the target sequence. Repeating this cycle, heat to separate, cool to bind primers, warm to extend, causes exponential amplification. One copy becomes 2, 2 become 4, 4 become 8. Within 30 cycles, billions of copies exist. What makes PCR so powerful is not merely its efficiency, but its precision. By designing the primers carefully, scientists can amplify only the DNA region of interest. Even a single nucleotide mutation can be targeted. And because Taq polymerase can withstand the required heat cycles, the entire process becomes automatic. A small machine, now ubiquitous in laboratories worldwide, cycles through the necessary temperatures without human intervention. This automation turned what used to be an enormous ordeal into a routine procedure performed countless times a day in research labs, hospitals, forensic facilities, and even at home during public health crises. PCR became the foundational method for modern molecular biology. Its applications are astonishingly broad. In medicine, PCR made it possible to detect genetic diseases quickly by amplifying relevant regions of DNA. Conditions like sickle cell disease, cystic fibrosis, and certain cancers could be diagnosed with unprecedented accuracy. PCR also transformed infectious disease detection. Viruses and bacteria leave behind genetic traces that PCR can amplify even when present in minimal quantities. This capability revolutionized the diagnosis of HIV, hepatitis, HPV, and countless other pathogens. In the realm of forensics, PCR ushered in the era of DNA fingerprinting. Criminal investigations, once reliant on blood typing or unreliable witness testimony, gained a technology capable of identifying individuals from minute biological samples. Cases were solved, wrongful convictions overturned, and forensic science established new standards of evidence. Archaeology and paleology also benefited. PCR enabled the extraction and amplification of ancient DNA from bones, teeth, and other preserved materials. Studies of human migration, extinct species, and evolutionary relationships became possible in ways that had once been unimaginable. In environmental science, PCR allowed researchers to detect species in an ecosystem based solely on fragments of DNA left behind in soil, water, or air. This technique, known as environmental DNA analysis, provides powerful insights into biodiversity, conservation, and ecological health. The development of Taq polymerase further refined PCR's reliability. Because primers bind more selectively at higher temperatures, Taq made the method cleaner and more specific. Unwanted background amplification, a persistent issue in earlier attempts, largely disappeared. The result was sharper, more reliable data. PCR did not emerge overnight. It came from a convergence of scientific efforts: the discovery of restriction enzymes, the isolation of Taq polymerase, the ability to synthesize primers, and decades of research into DNA structure and replication. The method was refined through collaboration, experimentation, and persistent problem-solving within the scientific community. As PCR matured, automated thermocyclers, machines that adjust temperatures at precise intervals, replaced manual cycling. These devices became staples of molecular biology labs worldwide. Portable versions later appeared, allowing field researchers to analyze DNA on site. PCR even played a central role in the development of vaccines and therapeutics. It enabled the study of genetic sequences that form the basis of many modern biotechnology innovations. In virology, PCR remains the primary method for identifying and tracking viral mutations. During the COVID-19 pandemic, PCR became a household term. The test that detected the virus relied on amplifying segments of viral RNA after converting them to DNA. Billions of people depended on this technology to navigate public health decisions, travel, and treatment. A technique once confined to specialized research labs suddenly became part of daily life. But the scientific meaning of PCR extends beyond its practical applications. It represents a profound shift in humanity's relationship with biology. For the first time, humans gained the ability to selectively amplify genetic information. This capability opened the door to genome sequencing, personalized medicine, and an entirely new branch of biotechnology. PCR exemplifies how scientific breakthroughs depend not only on ideas, but also on the right tools and conditions. The discovery of organisms in Yellowstone capable of surviving extreme heat might have seemed unrelated to genetic diagnostics. But nature often contains answers to questions not yet asked. The story also illustrates the power of cross-disciplinary thinking. Advances in chemistry, microbiology, engineering, and genetics combine to produce a method that none of those fields could have achieved alone. Scientific progress is rarely linear. It relies on unexpected connections, careful experimentation, and the willingness to imagine new possibilities. Today, PCR remains at the core of molecular biology, even as newer techniques such as CRISPR and next-generation sequencing expand the horizons of genetic research. Yet none of these would have been possible without the ability to reliably copy DNA. PCR is the foundation, the tool that unlocked the genetic code and made modern biotechnology possible. It is a striking reminder that the most transformative discoveries often come from simple ideas refined through persistence. Heat to separate, dot, cool to bind, warm to extend, repeat. A sequence of steps that mirrors the rhythm of biology itself. At Mythosium, we explore stories like this because they reveal how science advances, through curiosity, collaboration, and the relentless pursuit of understanding. If you enjoy discovering the ideas that shape our world and the technologies that define modern life, make sure to subscribe. There are countless breakthroughs left to explore, and we'll continue uncovering the ones that changed everything. Thanks for watching, and stay curious.